Good morning, everyone. Once again, I, Samresh Palroy, welcome all the esteemed participants across India in day four faculty development program on harnessing AI to revolutionize pharmaceutical education and research. Today, we have honor to welcome Dr. Swamiprakash Patel, an esteemed professor at Ramanbhai Patel College of Pharmacy, Charusat, Changa, Gujarat, with a PhD in pharmaceutical science and a rich professional journey spanning roles such as research coordinator and scientist. Dr. Patel has profound impact on research and innovation in the pharmaceutical field. Currently, Dr. Patel coordinates the patent facilitation cell and engage deeply in research coordination and teaching. He is registered patent agent, bringing significant expertise in intellectual property rights, formulation development, and novel drug delivery system. His notable achievements in the patent field include over 50 patent applications filed for clients, 5 patent application filed personally, 7 first examination report handled, 10 patent hearing attended, and also 5 patent granted for clients. So I think he's one of the resource person having a good experience in this patent filing and publishing. So Dr. Patel is also an active researcher with a focus on utilizing artificial intelligence for pharmaceutical science, having secured substantial funding and leading several high impact projects. So recently, sir has received one project under AI from GUJCOST, Gujarat Council of Science and Technology. Dr. Patel skills and encompass web designing, AI, machine learning, and much more, making him a versatile professional in the pharmaceutical education landscape. So, with this, I invite Dr. Patel to start his session on the topic implementation of text mining in patent search. Dr. Patel, please, you can unmute and start the session.
Thank you very much, sir, and thanks for providing this platform. So, should I share the screen?
Yes, sir. Yes, sir. Please, you can share.
Okay, okay, okay. Okay. Is it visible?
Yes, sir.
Okay,
So you can
okay,
on your
fine.
video
So,
also, sir. I can see you. You can just on your video.
yeah, just one minute.
Nice. Okay. Okay, sir.
And now? Okay.
Yes, yes, yes.
Okay, so I have made my presentation in full screen. I think it's visible now.
Yes, sir. It's totally
Okay.
visible.
So, welcome everyone. So today topic is development of how can we use AI in a literature survey. So today I will discuss with you what is the use of text mining specifically in a patent search. So basically text mining is not just for the patent search. It is also for the any kind of literature, right? So let's say if we are searching literature, non-patent literature, we can employ the text mining as well as in a patent search, books everywhere. So I will give you today a demonstration how we can perform a text mining in a patent search. So you can further do it by yourself with any kind of literature. So before we start with the demonstration, let me brief you what exactly text mining is and why how can implement and what are the basic steps of implementing the text mining. And demonstration I will give you with the software. Basically, text mining you can apply better with the coding, but many of the let's say people or fraternity from a pharmacy are not that much of the known to the coding, and they are basically not a programmer. So I will show you a GUI-based tool, so where you can apply the text mining and can understand your literatures. So what I'm going to show you today is a patent landscape. How can you prepare a patent landscape of the probiotic, right? So this is the just a brief. Then we can perform demonstration on any of the topic of the patent. So let's move further. Okay. So basically, if I want to search patents on a probiotic, so if I make a simple search on a probiotic, right? So you can see this is the literature, how the trend of the probiotics goes in the field, right? So it is a continuously rising and one of the let's say a boom topic nowadays. So this is how we select our topic for, let's say, our research or project whatsoever. And being a let's say pharmaceutics person, I am interested. For example, we are doing this as a, for example, you can employ text mining on any topic. So let's say I am interested in a formulations of the probiotics. So I perform patent search on certain tools. I have actually commercial tool Patsy, but I will show you a demonstration. How can you employ it on a free tools tool, right? So this is the numbers of the patents I found. This is 4,774 total patents. So let's say if you make a simple search in a PubMed or let's say in a Google Scholar or let's say Google Patent or any of the other platform for your topic, you may find so many results. And now you need to decide where exactly you should start. Rather, if you can employ certain filtering system, so you can exactly understand what is the entire picture of your topic, right? And then you can select the exact literature you should study. So here in my hand, there are 4,774 patents that definitely I cannot study all of them. But I need to first understand what is the exact picture of these patents. Means this is the all the patents regarding the probiotic formulations, right? So I want to understand first key con say what are the different formulations or what are the different types of technologies are covered in the patent. Now, how can I see that? How can I visualize it? How can I understand this or high, these huge numbers of the patent? So you can employ the tax mining. So basically, what is the tax mining? So tax mining is a kind of analysis, and it's the process of extracting useful information and insight from the large amount of unstructured data. So for example, if we have a numerical or digital information, right, we do perform some sort of the analysis, we do perform the statistics, and then we can conclude. Same way, when we say we have these numbers of huge literatures, right? So this is the actually data, but this data is in a text form, right? And this is unstructured text, means it is written lots of things in a different way. If you say patents or let's say research articles, so lots of things are written in unstructured way, right? So what does actually text mining do, and how can we implement the text mining? So first we have to do the just a minute. First we have to perform a data retrieval, right? So let's say if you want to perform a search on a particular topics of the patent, so you need to first search the patents in a particular let's say database, uh, and you need to export the data, right? That I will show. Then you need to perform a certain data pre-processing. Let's say if you just exports randomly and try to implement analysis on that raw data, right? So it will not help you, right? You need to pre-process data. For example, when you export the patent data, you may have a details of the inventor, you may have a details of the assignee, you may have a details of the application dates and kind of, which might be not of your interest, because your interest is to understand the technology, right? So, for example, right, so you have to remove this unnecessary information. So this is what you have to do. You have to perform a data pre-processing. Then you have to make up your final corpus preparation. So what is a corpus? Corpus is the final list of the text, right, that you have gathered to perform the further text analysis, right? So that the corpus preparation. Then we perform the text pre-processing, right? I will show you one by one steps for all of these, let's say, individual steps. So in text pre-processing, what we do, let's say, for example, some text is in a capital, some text is in a small letter. There are some grammatical words like and, or, and therefore, for example, in, so which are the meaningless text. So that we need to remove from the our data set, right? So we need to focus only on the technical terminologies. So this is called the text pre-processing. There are so many other things too, but we need to perform a text pre-processing. Once we perform a pre-processing, then we start our text mining, right? So these are the basic steps before we start with the text mining. So you have to collect the data, then you have to pre-process your data. You need to remove, or let's say simple terms, you need to just keep the information or text for which you need to perform the mining, right? Data analysis आप बोल सकते हैं in this case. Then you have to prepare your corpus, then you have to perform a text preparation, and then you finally perform the text mining to understand the technology, right? So here I have given the one tool, Orange Canvas, right? As I said, text mining can be performed on a many tools, right? Python is, I think, as per my understanding, and I use Python is the best language to perform the text mining, because it gives you lots of freedom to tweaks whatever your requirement. But let's say at the beginner levels too, and even at I feel at even let's say if you are a pro researcher too, this tool, Orange Canvas tool, is a GUI based tool. Basically, there is the Python script, but it is a GUI based tool which help you to perform the text mining very easily, right? So here I have shown you the flow of the uh, let's say text mining process in a Orange Canvas, right? So Orange Canvas is a free tool, and there are so many statisticals and machine learning based activity you can perform in a Orange Canvas. What you have to do in Orange Canvas is you have to first download Orange Canvas, and then you need to install the add-ons. There is the options in a software where you have to select the add-ons. So you select the add-ons for the text mining, and then add-ons will be installed, and then your platform will be ready to perform the text mining. Here is the flow. I have to first import the document. Then I need to fetch the text information from the documents. I can see the documents if I want to see. So there is one option called corpus viewer in the tool. Then I will perform the text pre-processing. Then what I'll do, I will perform tokenization. That's the that I will explain you when we perform the demonstration. So it will be better. But for now, let's say this one is a sentence. Let's say, for example, this one is a sentence. It involves using computational technique, for example. This is the sentence. Now, what this bag of words do, this bag of words and this text pre-processing tool will break this sentence into words. So it will break into each, then involves, then using. So ultimately, it breaks all of your sentences and then collect it as a words, make it transform it into words rather than let's say sentences, right? So this is the back of word. Then it will perform certain calculation, right now we are not discussing what exact calculation it is doing, but I will brief you what is the distance calculation. So basically, let's say there is a one patent where let's say probiotic. This is a probiotic formulation, for example. So let's say this is the one patent where the tablet based on the probiotic based tablets are prepared, and there is a one more patent where the capsules is prepared, for example, based on the let's say probiotic, right? So obviously this tablet word will be more time in this document one, and this capsule word will be more time in document number two. Now let's say there is a one more document or pattern, let's say three, where it is also about the tablets. Let's say it is something different tablet, different types of tablets, floating tablet or whatever it is, but it is a tablet. So definitely there are tablet words too. Now if we calculate the numbers of tablets occurring in this first document, let's say it is a 10 times, for example. Here numbers of capsule is occurring 10 times, and let's say there is some sort of description, and there is a one word of the tablet 2 in this document, right? And this is the third document where the tablet word is occurring, let's say 7 times, for example, and a capsule word is occurring 2 times, right? So now this distance tool, what it will calculate, let's say this document have a 10 times tablet word, this document have a 10 times capsule word, and this document has a 7 time tablet word. So these documents and these documents have a more similarity, and it has a more, or let's say less distance in terms of the numbers of occurring of the words, right? And there is a 2 times capsule words occur here, and there is a 10 times capsule words occur here. So these two document number 2 and 3 are comparatively far with each other, right? So this is how it actually calculates for the all of our let's say data, right? All of our, let's say if we are collected 4,000 patterns, so it will calculate this distance for all words in all the documents, right? So this is the tool what distance do. Then there is one more tool called hierarchical clustering. Now this hierarchical clustering will club the documents depending upon the technological terms, right? So let's say, as I said, this tablet word is occurring 10 times here and 10 times, 7 times here. So it will club these two documents in a one group. And let's say if any other document having a more words of capsule, then it will club it with this capsule, right? So this clustering is a tool where it will groups our, let's say, literature according to the context, actually, right? So if we say tablet is occurring more in this document, so definitely this patents or this literature is about the tablet formulation, and let's say capsule words is occurring so many times here. So let's say there are high probability this document is even regarding the capsules, right? So this hierarchical clustering will make a groups based on the let's say context of the documents, right? So here we can easily groups so many documents in a particular or last number. Let's say I will show you there are, let's say, 4,000 patterns, but it will divide that 4,000 into a 10, 20 or 30 groups, right? So basically there are 4,000 patterns, but all these patterns falls under the 30 common categories. So now I can understand that this entire let's say data set, or this entire, let's say, patents on the probiotic formulation, there are falls under basically 30 category, for example, right? So I need to focus on the 30 type of the invention, right? So there might be variation in these individual patents, but the overall picture I can understand thoroughly, right? So this is how we can perform the tax mining. Then we can individually understand the groups, what is the technology inside the group, by looking at the word cloud. So this is how we perform the text mining and then individual analysis, right? So this is about the text mining. So once you perform the text mining, this is what exactly uh, I told. This is the tool where you can perform the text mining. And once you perform the text mining, all these 4,000 patents are divided into these 20 groups. These are the different technological groups in which my entire 4,000 plus patents falls. If any of these groups shows me like there are more than 1,000 patents in a particular group, I can perform a further clustering. So it will further divide that 1,000 patent into further different 20 or 30, whatever the groups you want to understand and set, right? So this is how exactly we perform the text mining. So I have placed here certain uh, example here, right? So these are the tools of the pre-processing. हम इसको देखेंगे. We'll see in a let's say demonstration. So right now I'm not discussing here, but if you see what happens if you do pre-processing and if you do not pre-processing. So if you see the image of the let's say words. These are the word clouds before pre-processing and after pre-processing. So if you see in the left side, the word cloud of the my corpus without pre-processing. So there are so many useless words like and, the, of, or, right, in, is, which is meaningless, right? Very, which is technically useless because I want to focus on the technical terminology here. So once I perform a pre-processing, it will automatically remove these non-technical grammatical words. And if you see on the right hand side after pre-processing, there is the only important technical terms like this is a probiotic. So it will focusing on the terminology related to the probiotics like species, this bifidobacterium, for example, animalis, lactobacillus and all, right? So this is the impact of the pre-processing. We are going to perform a demonstration so you can understand easily. Uh, And once you perform this analysis, you can understand each group one by one. So let's say I have highlighted here the 5 groups out of the 20. So if you see the first group, right? So first group, the most occurring word is a traditional Chinese medicine, for example. So this is the group out of 4,000 patent where probiotics based on the traditional Chinese medicines are invented, right, or formulated. The second group is a group where the most occurring word is a beverage, right? Sorry, beverage. So this is the one group where all of the patents, all of the probiotic patents, they are the formulation of the probiotic, but they are focusing on the probiotic based beverage, right? If you see this cluster, so this one is the all the formulation let's say all the probiotic formulation focusing on the powder based formulation. This one is about the feeds of the animals, because probiotics nowadays is not for only being developed for the humans, animals and other species too, right? So if you see there are terminologies like pig, feeds, right, and other, let's say, chicken, right? So these are the livestocks, right? So these are the all the patents regarding to the probiotic formulations for the animals, right? And if you see, this is a very typical where the all the probiotics are developed, or let's say stability is improved, or the probiotics genetic structures are developed by employing certain acidic species and all. So this is how you can directly understand entire literature, and you can conclude ki basically out of this 4,000 plus patents on the probiotic, what is exactly inventors are doing, right? So let's say if you now want to understand more about the animal-based patents, then you can only focus on the this group, right? So you once you have an overall picture of the probiotic formulation, what are the different kinds of innovations are going on in a probiotic formulation, then you can further, let's say, lead and steer your research in a particular direction. But ultimately, what you did is within a very few clicks, you technically understood the technically gone through this 4,000 plus patents, right? So this is called the tax mining. Now, uh, I think, uh, yeah. So this is actually the groups I have listed here in my search. So let's say this is the animal feed preparation. How I come to a conclusion that this group is all about animal feed preparation. So in this cluster, I can see this type of the words like feed, pig, duck, fish, piglets, and all. This is about the traditional Chinese medicine because there are certain common words like medicine, Chinese, and traditionals. Then probiotic preparation with the antimicrobial activity. So in this group, I can see all these words like antimicrobial, strains, bacillus species, and all. Encapsulations are there. So let's say that in probiotic have an issue with the stability. So here are all the innovations where the probiotic strains are encapsulated with the different technologies, right? And here is the probiotic foods, because there are common occurring word like food, milk, bread, and chocolate. So this is how this tax mining divide, let's say, make groups of your 4,000 patents, and you can now easily understand. Let's say this one is the only words where arthritis. So all of this invention, all of the probiotic formulation focused only on the arthritis, right? So you can, let's say, directly understand if you want to further study what are the different probiotics innovation which works in the arthritis, so you can directly select these words and can only focus on that pattern. So this is, I think, enough to understand a very let's say, to have a very brief idea what exactly the tax mining and what exactly it does. These are the other let's say groups. So this is the brief of the tax mining, and now let's move to the live demonstration so you can exactly understand and can note down steps, right? So you can further perform it by your own. Okay, so I think I have uh, switched my screen. Is it visible?
Yes, sir, it's visible, sir.
Okay, so let's first start with the patent search. I have my topic, and if you want on your topic, so that's also fine. But let's first do with my topic, so then you can understand exactly what's going on. Then you give me any of the topic. So let's say we have a Paxil here, but I will do it on a open source platform, lens.org. Right. So I think there are so many platforms where you can perform the patent search. But I do believe this lens.org is the best one, because on a let's say free platform, it is also partially paid and partially free. But in a free platform, it allowed so many functionality which other platforms are not allowing in a free platform. So let's say if you have to perform a lens, if you have to perform patents search, Google Patent is a one tool, but the Lens is one of the best in a, let's say, free tool. So I will not directly make a search here. What I will do, I will go in a structured search, right? I will click the structured search. Why structured search? Because now see, when you are collecting your literature, you also need to do some sort of background kind of homework. Means, let's say, if you are working on certain technology, you have to do certain background homework or basic understanding about the topic, right? Let's say if we do a patent search, it is always advisable to do a patent search in a title, abstract, and claim, right? So basically, patents में क्या होता है कि this title, abstract, and claim component of the patent, like in a literature, like in our research article, there are certain segments like title, abstracts, then introduction, material, method, result, discussion. Likewise, in the patent, that these are the different sections. But this title, abstract, and claims are bound with the Patent Act, with the rules. There are rules. You cannot write anything. You cannot write vaguely in these three sections like title, abstract, and claim. Let's say if I am uh, preparing a patent document about tablet, then I can write anything about, like, say, in a description, I can write the disadvantage of the parenteral preparation, disadvantage of the syrup suspension, disadvantage of the capsule. So that will be written in the description, right? So if I search tablet or capsule in a description, there are so many chances that I will get the wrong document. Okay, because inventor have written a, let's say, background studies, background, or let's say history of the tablets and all in a description. But if you search in a title, abstract, and claim, where as per the Patent Act, you have to focus and write uh, only about your invention, right? So it's always advantageous and advisable to search on a title, abstract, and claim section only. So let's first try with the, let's say, I will try with the malaria formulations, right? So I have written here malaria formulations, but this search portal, always this patent search portal is like Google search. If you search malaria and formulation in a Google, so you may find so many documents about the malaria formulation, but you may also find so many documents where this malaria only malaria word is occurring, and so many documents where the formulation only words is occurring. And my focus is only patent technology or patents which is based on the malarial formulations, right? So what I'll do, if you see search tips, now this is something called the patent search techniques, and definitely you need to acquire certain this patent search skill uh, by your own. By, let's say, there are so many other workshops and courses you need to do. आप YouTube पे भी देखेंगे तो बहुत सारे आपको वीडियोस मिल जाएंगे how to do patent search. But if you see here the search tips, there is one option called the proximity search, right? Proximity search. So what is proximity search basically? So proximity search is about this malaria and formulation. Both word I required and both word be near to each other. So let's say if the invention or the document, let's say specifically is about the malarial formulation, so definitely this malaria and formulation words occurs nearly in a one sentence, or let's say at least in a one single paragraph. So that's why I do perform the proximity search. So let's say I want that this malaria and formulation both word must happen or must be within the proximity of the 5 words to each other. So it will increase the probability that the document I'm getting is mostly on the malarial formulations, right? Because there are so many chances. If I just simply search malaria and formulation or malaria formulation, it will give me a document where the malaria word occurring only and the formulation words occurring only. Or I may get the malaria and formulation too. But at the end, I will have so many unnecessary documents. So this is the one trick. So this is the actually patent search trick. If you are performing a simple literature search, let's say for example article search and all, then you don't need to do this kind of things. I will show you how can you do it in a software tool. So here I am searching. So the first option, first step for the text mining was like to collect the database, to collect the data, right? So here I'm actually collecting the data. So here, if you see, I have a patent record, 1360 patent, which I can never read, never read. I cannot understand 1360 patents as such. अगर मैं उसको पढ़ना भी स्टार्ट करूँ टाइटल भी, या फिर आई जस्ट कलेक्ट द टाइटल, आई विल शो यू. Collect the title, you cannot understand what exact picture behind this 1360 patents, right? So let's first export this record. So here is an option called export. So this is what I'm telling. Lines.org is giving the so many functionality in a free, let's say, tool, as a free tool, which other platforms are not providing. So I will export. Once I click the export, you may see you can export at a once 1000 patents in a format. So you can change the format, but the CSV is the best format to perform the text mining. So CSV is nothing but a comma separated value, and it is as simple as like Excel. You can open the CSV in a Microsoft Excel too. Now, what are the field it will collect? It will collect the so many information about this 1000+ patents. But my interest for the text mining is a title and abstract. Title and abstract. So these are the most important tags which allow me to understand the patent, right? But right now I'm collecting all the information, so we can do certain more analysis too in a orange canvas. So I am just simply clicking export. Now it will export the document. And now it is exported. So it is now exported. I'm opening it. So as I said, this is a .csv format and you can easily open in an Excel. Okay, so this is my data set, right? So if you see, this is number of the documents and it is more than 1000. If I select here, you can see the numbers here, and 1001. The first one is a title. So there are 1000 patents here. This is about the jurisdictions, right? So कौन से country का patent है वो है, कौन से stage पे है वो है, application number से, lens का खुद का ID है, date of publications वगैरह है, and there is one title and abstract. So if you see the titles, right, here are the titles. Some are in a capital, some are in a sentence case, right? Some have used dash and all. So if you try to read these all the titles, even you read the titles, you cannot understand what exactly going on here, right? What is the picture behind the material formulation here, right? So you cannot understand. So what we'll do, here is the text about abstract of this patent, particular patent. So what we'll do now, we'll perform the text mining and try to understand what is the technologies or what are the common domains in this all the patents falls. Okay, so I'm closing these CSV documents. Now already we have collected data, so I'm minimizing it. If I'm doing it. So now I'm opening the orange canvas. I have already installed. Sir, am I clearly audible and all? All the audio visuals are going well.
Yes, sir. Yes, it's clear.
Oh, thanks. Okay, so when you open your orange canvas,
So can you please be some more loud?
oh yeah,
So
sure. I think now is it clear?
yes, sir. Now
Loud
it's
enough?
loud. Yes, yes,
Okay,
yes.
okay. So when you open this orange canvas, it will allow you to create a new document, but basically already there is a new document. So
It
you
is free
don't
tool,
need to,
sir. Orange Canvas is free tool.
yeah, it's a completely free open source tool, sir.
Okay,
Completely
okay,
free,
okay. From
right?
directly from Google, you can download.
You can Google download, you can cite
Okay,
wherever, let's say in your research manuscripts and all. It's a completely open source documents
okay,
software.
okay, okay. Okay, thank you,
Okay,
sir.
so you have to just close it. Now this is your white space, and here you can see there are different tools. So as I said, these are actually GUI based software. So whenever you select any tool, background में एक Python script run होता है, right? So this is a Python based tool actually. So once you install it, when you first time install, you may not have this much of the add-ons. You may know how you will not have any of the add-ons. What you have to do, you have to click on the option and you have to click on the add-ons. When you click on the add-ons, you will have an entire list of the add-ons. So whatever the add-ons you require, you have to click and just press OK. So it will download and it will install, right? So for our text mining, we need a text and text table, these two tools, right? So I have actually installed all the add-ons. So if you see, here is the entire section of the text mining. Here is the text mining. So these are all the tools of the text mining, right? And you can, there is the one tool called the PubMed tool. So directly you can search PubMed documents from this software and can perform the text mining analysis directly. I will also show that, but let's first focus on the patent today. What I will start with the file. So when I click the file, so there is a one let's say button here called the file. Now when I make a double click on it, right? So here it will allow me to select my document. So whatever the document we have exported, the text document, right? So I will collect it. So this was the lens export. I have made so many, let's say, trials before this session. So let's say this is a lens export 8. This is about all the 1000 patents on let's say malaria. So I have selected it here, and now I will uh, close it, right? So here is my file. I have given it a file. Now, how can I see this file? So when you stretch it, right? This is how this software works, right? So you have to select and just stretch it. Just stretch it. And now all the tools in my left-hand side panel can be accessed here, right? So what I will do here, I will add a document, sorry, uh, just data table, right? So basically it's an Excel, right? So I can show you, right? So when I open it, so these are the all the 1000 documents, right? These are the all 1000 documents. All the information I have just shown you in Excel is now visible here too, right? So this is all my document. Now, if you want to see the basic information, so here you can see, sorry, yeah, one tool called data info. So if you click data info and if you connect this file to data info and then open it, so it will give you basic information. So what is it? 1000 raw. 1000 raw means there are 1000 data. There are 33 total columns, right? So there are 32 different types of information in our data set. 5 categorical data, 12 numerical data, and 16 text data, right? So this is how you can easily understand what is exactly your data set, right? So I'm keeping it, sorry, I'm putting it aside, right? So now what I'll do, uh, I will select columns because there are so many columns and we are only interested in a text uh, that is title and abstract. So I will choose this option called select column, right? या फिर आप कर सकते हैं कि आपका जो एक्सेल है उसी में से बाकी का जो डेटा है वो डिलीट कर दीजिए, सिर्फ दो ही कॉलम रखिए, टाइटल एंड एब्स्ट्रैक्ट एंड देन गिव इट। यहाँ पे मैंने सारे रखे बिकॉज आई वांट टू शो यू वन मोर टूल्स ऑफ दिस ऑरेंज कैनवास, लेट्स से पाइवोट टेबल हम लोग एक्सेल में भी करते हैं, तो दैट्स फंक्शनलिटी इज हियर। So if you select the pivot table, it will allow you to understand your data statistically. ठीक है, तो इसमें देखो मैंने there are as I shown you कि बहुत सारे फील्ड है, एप्लीकेशन डेट है, एप्लीकेशन ईयर है, अर्ली पेमेंट टाइप है, फुल टैक्स है या नहीं वो डेटा है, राइट? So here I have selected jurisdictions and its legal status, right? So here I can see these are the juris sections. So out of 1000 पेटेंट, ये 5 कंट्री या 6 कंट्री जो है, फॉर एग्जांपल कनाडा, चाइना, यूरोप है, this is I think something European country, New Zealand, US, and this is WIPO patents. So these are the 5 6 jurisdictions में ही ये सारे patents आ गए, ठीक है? उसमें से if you see here the highest numbers of the patents are from the US, 457 है यहाँ पे। WIPO patents means international patents जो है वो 410 है, then European patents and all, right? There is no any Indian patent. मे बी चांसेस है कि lens.org जहाँ से डेटा में लिया हमने फ्री सोर्स में से, उसमें देर आर सर्टेन रेस्ट्रिक्शंस, दे डू नॉट गिव ऑल डेटा from the let's say free access, but let's say if you have access to a commercial database for the patent like Patsir and all, so you will have an entire data set, right? So this is how you can perform, let's say how many of these documents for example are having a full text, right? So out of these 1000, there are 995 documents are the full text. So we can access the 995 documents out of 1000 in a lens as a full text document. There are 5 documents जिसका maybe abstract या फिर limited information available है. It's the Canadian and Chinese patent, right? If you this European and New Zealand patent. So this is how you can perform the statistics here, right? You can perform graphical analysis too. This is not a let's say part of today's session actually, but I will show you हमने tool open ही किया है तो let's say scattered plot for example. So here you can perform a scattered plot. So again legal status and jurisdiction. So these are the active patents, these are the discontinued patents, these are the expired patents, right? So if you select this only active patents, if you select only active patents and then try to understand the data table, what are these active patents? So it will show you only active patents. So see out of 1000 patents, there are very limited, 158 are the only active patents, right? Others are the either expired, either discontinued, kind of, right? So as I said, you must have a basic understanding about your literature survey, whatever you are doing, right? Okay, so I'm deleting actually unnecessarily. I'm not making this, let's say platform bulky, which is not today's session. Today's session is about the text mining. Okay, so here what I'll do, I will collect the tools, collect this. So here is the list of the columns. As I said, there are 33 columns. So this is all the 33 columns. And this dark one is something text. This green one is something about the categorical. And this blue one are the digital data, right? So out of these are the ignored columns, right? Here you can see these are the ignored columns. So I have ignored because I don't want to perform a text mining on these columns. I just want to perform a text mining on title and abstract. Now, how can I do that? Simply just you drag and drop wherever you want to put it. Let's say if you don't want the title, you just put it here. I want the title, just drag it and put it here, right? So simply it will perform a certain will run certain script in the background and it will select only title and abstract, right? So I have selected title and abstract only. Now what? Now I will consider this data as corpus, right? So this is my corpus. Now corpus is nothing but the text data. It's a collection of the text data. So if you see here corpus viewer, if you put it here like corpus viewer, so it will show you the selected documents. So, right, these are the 1000 patents, right? We have selected just title and abstract. So now this is my data. It has selected only title and abstract. Like some patent may not have, let's say, abstract, for example, here, right? So whatever it is, but it has collected text data, title and abstract for the all entire literature, right? So you can see the corpus too. Now this is my data. Now corpus is the collection of the text data. Now I will perform the pre-processing here. So if you add a pre-processed text, so here now this red dot means script is running. So you need to wait, right? Okay. So now analysis is performed and here is the pre-processing. Now let me show you what exactly happening here one by one. Just let me zoom my window if I can. Okay, is it? आपके एंड पे जूम हो गया सर?
Yes, sir. It's Zoom.
Yeah, okay, okay.
Ah,
You can, everybody can see it, right? Okay. So what's going on here is pre-processing. So exactly what I'm doing, let's say for example, transformation. So what I'm doing, if you have seen the text, some of the title in capital, some of the title in the small letter. And as I say, this is the natural language processing, NLP. NLP is actually part of the one type of the AI. मशीन लर्निंग के बहुत सारे डिवीजन है उसमें से नेचुरल लैंग्वेज प्रोसेसिंग तो बेसिकली what exactly it doing, it is collecting the data. Now if we feed for example tablet word in a capital and tablet word in a small, it will consider both the word as a two separate word because the system itself do not understand these two words are the same actually. So we need to perform certain pre-processing. For example, I have transferred all the text into the lowercase. If there is some sort of any accents and all, so I have removed it. If any HTML or something links are there, so it has passed it and collected the text from it. URLs are there in my data, so it is removed. So that's the first step of the pre-processing called the transformation. So I have selected all, and this is by default you should do, right? At least in the biological science literature text meaning. Next is tokenization. Now, what is tokenization exactly? So tokenization means I told you that we have to break the sentences into words, right? So now what this tokenization, there are different types of option and sometimes you need to try, you need to perform trial and error, right? This I have selected this REGEXP. REGEXP means a regular expression. Now, what is it? Slash W plus, right? So what does it mean? P, whenever it finds a white space in the sentence, right? So when we find the white space we find between two words, right? So whenever it finds this white space, so it will break it, right? So it will break all the words. So this is the best tokenization method, right? Uh, then next is a filtering. Filtering. This is very important. Now I have selected these few options. So first one is the stop word. Stop word of the different languages. I think most of the document I have collected are in English, right? So I have selected English as a stop word. So what it do? It will filter out all English non-technical words as I shown you in a presentation, right? Like in, for, and, right? These all predicates words, meaningless words. They have a meaning in definitely in a grammar, but they don't have a importance, let's say technical context of the, let's say this analysis. So I have select this English dictionary and it will remove all of the stop words. We can also manually add certain words that I will show you. Right now, I am keeping it to none. Let's say, for example, this is a patent. So definitely invention, novel, then unique, patent, claim, disclose. These are the common words of the patent documents, right? Let's say if you are searching with the malaria here, for example. So malaria is definitely it's a common word. It will appear several times. So we don't want to focus on the malaria word because we already searched for the malaria formulation. So later what I will show you, I will put the words here like malaria and all, and it will filter out. Then the lexicon. Lexicon is something that you want to keep, right? If you want to keep something. So here again, you can create a list. Normally in a text document notepad file बनाएंगे notepad file पे जो जो words आपको चाहिए that you don't want to remove, right? Let's say by performing certain 50%, 60% analysis, you come to a conclusion that these are the common words I should keep, right? So you can make a list and you can keep it here. Then numbers. So numbers I don't want. Let's say focus here. So it will remove the numbers. Then it will remove all the regular expression like slash, dot, spot. For example, full stop, comma, semicolon, question marks, double inverted comma, and let's say any of the others you can add. You can write any. Let's say if you want to add, you can add here. It will remove, right? Uh, then this is an n-gram range. Basically, we put just a 1 1, how it breaks actually, right? Let's say we eat is a tablet. So let's say we want to break each single words, right? So if we keep two here, so it will consider it is as a one word. So it will break it is as a one word. Then again is as a one word and a tablet. So generally it's a very common in we keep it one by one. So if you are performing this analysis and keep the things by default here, if you are opening this pre-processing and keep the things by default here, then also it will work. If you don't have any detailed understanding, and if you keep it as the default, then also it will work, right? If you want to tweak something afterward, then you need to understand each and everything. Basically, it is already given in the website of the Orange Canvas, so you can understand. And these are very common terminologies and common tricks and tactics in the world of the text mining and AI, right? So I'm closing it right now. So this is how I have pre-processed. Right now, my pre-processing completed. Now I will collect the words, right? Because pre-processing has broken my sentences. Now I do not have things left. As a sentence, I have just words. So let me show you how exactly it looks like. So now if you see, uh, yeah, if you see, now let's say this is the sentence compound derived from the artisanates, right? So here it is how broken in the word compound, derived, artisanates, preparation, process, pharmaceutical. These are the individual words now, right? Based on my the pre-processing. This is in each document. This is in each document. So now my text data is now broken and now it is a simple a bag of words, right? So let's delete purpose here and now let's connect it with the distance. So as I said, what the distance exactly do? Distance will calculate. It will basically it's a very big calculation. It will create an entire matrix. Which words is present in which documents, how many times? That will it calculates, right? So that's a distance calculation. Based on that, it will calculate the distance between the two documents, how two documents are near to each other and how these two documents are far to each other. So this is how it calculates. And accordingly, it will make a clustering. So here you can see here, these are all documents, right? You can see here the titles and it has grouped them based on their common technologies, right? So now here you can have access to change, right? Let's say this is here, I have selected top 20 group, and let's say I just want to focus on top 10 groups. So it will focus on top 10 groups. Manually, if I want to, let's say, change it, so it will change, right? So it will change and it will give you more and precise grouping, but definitely numbers of group will be increased. So at the cost of numbers, let's say here there are 1,000 patents, but let's say if I want to do a very regressively, I want to understand this pattern, then there are 83 groups here, right? But let's say I want to overall understand the picture first, so I will simply divide them in a 20 groups, for example, here. So now based on this words occurring, now it is divided into 20 groups, right? So now I will see these 20 groups individually, one by one. So here is my groups, 20 groups. Here all the patterns divided. Now, how exactly it looks like. So this one, if I select, it will give us all the information about the first cluster. So basically this all 1,000 patterns are divided in 20 clusters, right? So how it looks like. So if I select the word cloud for the first cluster, it looks like this, right? And if I see the most occurring, so if you see here, there are 120 plus documents in the first cluster. And if I select this first word method, right? So out of 126, 119, let's say patents or documents have this common word method and composition. If I select, almost all the documents have these two common words, right? Right now I have not added any filter word, so we are looking it right now. Then we'll make it we'll fine tune more, right, by adding the filter words, right? So this is the first cluster. If I select the second cluster, so here you can see the compositions, right? So these are all about the pharmaceutical composition. My patent is on malarial formulation. So this is the let's say cluster. If I select the composition and pharmaceutical composition, out of 62 patents, 61 patents have these words. So definitely these patents are falling under this category of pharmaceutical composition. If I select the third cluster, for example, so this third cluster is focusing on antigen. If I select this word antigen, so 25 out of 25 document of antigen. So this all the patents are talking about the malarial formulations where antigens is involved, and this plasmodium word is also there. There are 20 patents for the plasmodium and there are 19 on the falciparum, right? So if I'm interested on the antigen based formulation, so I should focus on this only, right? So this how one by one you can understand. Let's say this cluster is talking about the vaccines, right? So this is the malarial formulations, but this is the formulation regarding the vaccines. How can you see that? Let's say, let's say I select this word vaccine, right? And let's say protein. Mostly vaccines and proteins are connecting words. And then I will see the corpus viewer here. So they will show me these all the patents. So here you can see isolation, purification, falciparum mazorite protein, 142 vaccines. So this is all about the vaccine patent composition, right? For example, this is a C vaccines, right? And all, right? This is the one protein and vaccines, right? So let's say you can further make a more cluster from the you can recluster it. You can break every cluster and perform the further detailed analysis too. So likewise, let's say three. So the third one and this C4 and C5 look similar. There are vaccines, but this is the pure. If you see, if you click on the C4 and then if you click on the vaccine, there are only 21 documents. But if you see C5 and then click on the vaccine, the 40 out of 40 documents are of the vaccines, right? So this is how one by one you can understand the all the, let's say, the picture, right? Now, let's say, let's make a certain filter because we have made certain things like this is a patent documents, right? So what I did here, I have just created one notepad file, and you can see here the patent, innovation, invention, claim, new, novel, unique, thereof, composition, then method. If I see so many method things here, so definitely it is the process patents, right? So instead of the process, they are using the word method here. So I have placed the method because this is a, after just making an overall picture, I come to a conclusion that method is a meaningless words here. The method use, then treatment, formulation, malaria is definitely, and formulation is definitely, it's a common word. This is the malarial formulation and disease. So I have made this list of the word. Now what I'll do, I will make a filter here. So what I'll do, I will add into a stock word, right? So I will select this filter .txt notepad file, and now everything again happens recalculated again, right? And just make sure there are 20 groups. Right now, if you see these numbers and everything analysis is different. Now, if you see the first cluster, so this is about the small molecule and boron. So this is the patent on the boron. All the patents are on the boron containing small molecule, boron containing small molecule. So this is the, you can understand, this is how you can understand one of the segment of the malaria formulation is a boron based small molecules, right? So these are the all patents on the boron based. Let's say second cluster. In the second cluster, these are the all anti-malarial agents. You can see anti-malarials and agents. So all are the anti-malarial agents. If you select these three words, 30 out of 30 documents are on anti-malarial agents. Again, this is anti-malarial agent, malarial, anti and malarial. Both word might be written as a common. So you also need to remove anti-malarial if you are not interested in anti-malarial agents. Then let's say this is third clusters. These are the compounds, heterocyclic compounds, right? Then therapeutic agents, all are the therapeutic agents is 55 or 55. So maybe therapeutic agents is a, it looks meaningless here. Definitely, if it is a malarial formulation, so all are the therapeutic agents, right? So you can easily remove it, right? So you just make a list, edit the list in a filter here, and then uh, just add here, let's say, for example, therapeutic agent, or let's say simply I'm putting here agent. So it will filter out this all the words. Anyway, but this is how you can easily understand 1000 patent just by the clicks. What is exactly, what is the entire, let's say, overall picture of these patents or technologies means how the inventors are exactly working in a different domains. So that you can easily understand without reading each documents individually, right? That is practically impossible. Now, as I said, this is the patent document, but let's say if you want to make a non-patent document, let's say for example, there is right now in Orange, there is a functionality of the PubMed only. So let's say I'm using the PubMed. So let's say you have to first write the email. So I have added my email. You don't need a password actually. Then let's say here I am writing malaria formulation and then find record. So definitely malaria and formulation have so many records. So you can see there are 2289 documents here. Let's say I collect, for example, uh, I'm not right now collecting all the documents. You can collect all the 2289 documents, but definitely it will take time. So right now I'm collecting just 100 documents, right? So I will retrieve 100 documents. You can see here the process has been started. So now all the documents are collected. Now, instead of this file, what I'll do, I will just disconnect this file and I will connect, sorry, remove this file. I will connect, uh, let it happen. So this processing is going on. So that's why taking its time. I will connect this as my corpus because this is directly the collection of, if you see, I have just collected the article title and abstract. I have not collected all details, right? So it is directly titles in abstract. So now if you see, this is the how the 100 documents you can easily understand. So these are the malarial formulation where they are talking about the vaccine. These are all the papers they were discussing about the vaccine. These are adjuvants. These are, let's say, for example, Plasmodium, Vivax, right? These are the documents which are focused on falciparum and Plasmodium. So you can club these both the clusters to understand, right? And at some point, if you think that you require a detailed clustering, so you can again, as I said, you can make a different clustering. So here you can see almost clustering is fine. There is no many documents. Sometimes what happens, so many documents, 100, 200 documents falls under the single cluster, right? So in that case, you need to recluster it. But if you see here, each cluster have a 5, 10 documents, right? So this seems proper and let's say detailed grouping, and that you can one by one understand. So this is all about the text mining. So I think from my point, my end, I think I have discussed a lot thing on a tax mining. This is actually the application based discussion. Definitely you need to individually understand what are these different tools, because but they are the part of the AI, so we are not discussing that much of the detail. Let's say if you change this normalization from this lemmatizer to the wordlet lemmatizer, so that algorithm will normalize the sentences and all in a different way, and you might have a different and better grouping, right? So you should try basically how exactly your corpus is, right? You cannot say every time that this normalization is perfect. This normalization based on the corpus that you have selected, you have to optimize the normalization, optimize the soft words and filtering, right? So once you did do that, you can understand n numbers of the literature at once on a one screen. uh, this is about the text mining. Let's say if you want to do it on your topic, I can do it again. Otherwise, I have other things too, as actually sir ने मुझे suggest किया था to showcase the other works that that actually works on the let's say AI technology in pharma. So we can take a very short break of 5 minutes, and then I can explain you, I can show you what are the different other works that I am working with the AI in the pharmaceutics. And again, this I'm open for the questions if you have.
Participant, if you have any questions for up to this, you can ask. So we can go for a 1 to 2 minute break after the question answer round. Again, we'll restart. Anyone like to ask anything? Yes, I can see Ashwini Mishra. Dr. Ashwini, you can please unmute and you can ask. Mhm.
Sir, am I audible to you?
Yes,
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 sir.
you're audible.
Actually, sir, this canvas what actually you have just shown for data mining. Apart from this data mining, sometimes it happens that if you are using certain title for even design patenting, then uh, there are slightly difference into the words and it might got accepted. So, is there any way how could like we guess that what could be that that word difference that could be identified, and on the basis of that we can go for patent filing.
Definitely, sir. Sir, patent is a different scenario. Exactly. You said ki if you make a certain tweaks, because patent always based on the document, you don't need to submit any proof. Let's say if you are working on a tablet or any technology, you don't have to submit that technology to the patent office. Everything is being examined on the documents, right? So let's say if you make the certain tweaks, if you are smart enough and let's say if you make a very rigorous patent search and you draft your application in a such a way that examiner will not find anything resembling to your, let's say, wordings, or resembling to your, let's say, innovation that you have written just on document, just by tweaking and replacing the word, definitely it will go. And but how you'll use it? Patent is basically, once you have granted, then you have to commercialize it, you have to deploy it in a real world. If it is a fake, then definitely you will have a points and granted one, but you cannot use it. It's useless.
Okay, no, actually, I'm looking for the real patent only, not for the fake one. But sometimes what happened that due to minute variation into the what do we call it title or the language, sometimes you may get stuck, and due to which like sometimes this problem occurs while filing it. That's why like I was asking the question. Thank you very much for your support, and it was really a nice presentation. Thank you for it.
Thank you.
You, sir. Now I like to request Dr. Pankaj Narkar. Dr. Pankaj, you can unmute.
Yes, good morning, sir.
Good
Uh,
morning, sir.
Actually, I want to ask more about the orange that just now you have described. Because I feel that if you practice this software more, you will get experience to use particular things. So, is there any particular material available for the more practicing this orange?
Yeah, definitely. If you see, visit the Orange website, they have very finely described everything, every tool, and they have also developed the very short YouTube videos for each tools they are developing. They are, let's say, 4 years back, they do not have that this much of the tools, right? But now they are adding more and more tools, so they are preparing short videos. Everything is on their website. If you go and search, but definitely you also need to perform certain, let's say, आपको भी थोड़ा study करना पड़ेगा, let's say, for each and every techniques, right?
Yes.
But how to do is given. How to do is given. Why you need to do certain background research?
And
Hope you are received the answer. Now I like to request Dr. Geeta. You can please unmute and you can ask your question. Dr. Geeta.
Sir, good morning, sir.
Good morning.
Thank you for your excellent presentation. Sir, actually, I have got a patent for one work, sir. Pharmacy, I'm from pharmacy department. So, any guidance to go for commercialization, sir?
Commercialization is always difficult, but
Yes, sir.
not, let's say, impossible.
Yes.
There are so many government platforms where you can apply. There is a one platform of WIPO called TISC, TISC
Yes, sir.
centers. TISC centers is there. You can contact to the TISC center. They have a, let's say, very huge pool of the, let's say, industries and all.
Yes, sir.
And definitely you have to work, you have to meet the people, you have to showcase your work in the different platforms. You need to search who are exactly in the same domain they are working, where you can approach. But I do believe that the best way is to commercialize by your own. If it is a
Yes, sir.
I don't know what is it exactly, but
Mhm.
rather finding someone to buy it from you, why you
Yes.
cannot start your own startup or something. I don't know what is exactly product process it. But
Yes.
yeah, one is a TISC, then one is a NRDC, is one government agency.
Yeah,
They
yeah,
also
yeah.
help.
Actually, I have got patent from NRDC, sir.
Yeah. Okay.
Yes.
So NRDC people also try to, but this is, let's say, a bit tedious job. You have to mail and contact everyone. You have to showcase your technology. You have to prove that this technology can be, let's say, executed at a large scale, commercial scale. If everything goes well, then they can buy and all.
It's a
So,
huge process.
right.
Thank you, sir.
Thank you, ma'am. So everyone, now we can go for a 5 minute bio break. So after 5 minutes, we will start the session. Now the time is 11:16. By 11:21, we'll restart.
Okay.
Okay, so we'll go for a 5 minute bio break. So welcome again after the short break. So I request again, Sir Swamiprakash sir, you can please start your session.
Okay, sir. So once again, welcome to everyone. So I'm going to show you how exactly a few of my works in AI. So yeah. So right now I'm working on few projects. So basically, if you see the AI in a healthcare, so there are so many things going on. Diagnostics, drug discoveries and all. But I mostly focus on the formulation, or let's say statistics segment of the AI. So here are my different works. I will show you few of them. Let's mm, yeah. Okay. So this is one of the work which is just recently published in IAP. So we have developed one tool called DE Interact, and it is in open domain. So you can also open it, DE Interact. What is basically DE Interact? So let me show you. So I'm just skipping how exactly the things works in ANN. Basically, if you have heard artificial neural network, it is something similar to the our human brain, right? So let's say, for example, if you consider a human brain, so what's a human brain is? So let's say it's collect the information from the neurons, right? And then here this information is stored and processed, and then it will let's say disseminate the response, right? The same way these mathematical, basically artificial neural network is nothing but a kind of algorithm. So here we give the inputs, then we'll trial uh, algorithm to change these weights. Weights is are nothing but the random numbers. So it continuously change these numbers. But we tell it that like you change this number, so ultimately it will gives the exact output for that we desired, right? So once it uh, uh, attain these exact values of these numbers, right? These weights, uh, so we can use that algorithm further for giving the input and predicting the something, right? So this is called the artificial neural network or machine learning. So what we have used here, so we have developed a artificial neural network machine learning based tool to predict the drug excipient interaction. So let's say in a pharmaceutics, when we are developing any formulation, we need to check whether our drug is compatible with the certain excipients or not, right? So what we have to do, we either have to perform certain analysis like DSC, XRD, या फिर you have to perform a real-time stability study of drug with a particular excipients, right? You need to store it at let's say high temperature, accelerated stability study करना पड़ेगा वगैरह वगैरह. Rather, what we did here, we have collected the data from the literatures, confirmed the literature, uh, where let's say, for example, this drug is incompatible with these excipients, and this drug is compatible with this excipient. So we have collected data. Now we have collected very huge data. So you see here, somewhere it might be the written. It's not written here, but we have collected something 1700 plus data. We have amplified it. So we have ultimately 3,000 plus data set. Then what we do, we have transformed our drug and excipients into fingerprint, right? So this is something a chemistry part. So we have transferred, let's say, for example, here is the aspirin. So how can we give, let's say, system, the molecule aspirin? For example, aspirin is incompatible with the polyvinyl pyrrolidone. So how can we feed this data? So data must be in a numerical form. So how we transform this structure into the digits. So this is called the fingerprint, right? So this is the PubChem fingerprint. PubChem is a very huge database of the compounds, right? So this is a PubChem fingerprint, and this is a very unique fingerprint. So if you see, this is a 881 digits binary fingerprints. So it represents 1 and 0. So shortly, if I say 0 to 114. So let's say if I start from here to here, for example, this first 114 numbers or positions is representing the elements counts. So let's say, for example, if carbon is present, so on a first position, it will give 1. If it is not, then it will give 0. Then let's say if it's nitrogen, so if it is present, then on a second position, it will give 1, otherwise 0, kind of hierarchical element. Then 115 to 262, let's say, for example, this much of the positions, right? So this is about the rings. So for example, 1 ring present, 2 ring present, heterocyclic ring present or not present. So based on that, it will give 0 and 1. Then single atom pairs, simple atom nearest neighbor pair, detail atom pair. So ultimately, this fingerprint represents the entire chemistry of the compound, right? Which group is present, hydroxyl group is present or not, carboxylic group present or not. So these all are covered in this fingerprinting. It is a unique for each compound. So we have transferred as our data set into the fingerprint, and then we have designed our data set in such a way. How? So let's say, for example, this fluoxetine is one drug and lactose is a excipient. So this is the fingerprint of the fluoxetine. So as I said, this is 881 bit fingerprint, PubChem fingerprint, 1 and 0, 1 and 0. Then this is a lactose fingerprint, right? And let's say, for example, these two compounds are incompatible. If our, let's say, literature, detailed study books, right? If it is incompatible, you may find so many examples in, let's say, internet, in literature, that these two materials are incompatible. Basically, it's a primary amine, so you know that it's a Maillard reactions and all, right? So this is incompatible. So I have written it as the incompatible as my output, right? So in input, what I give, I give the chemistry of the compounds, chemistry of the drug, and chemistry of the excipients. And in output, I give these two categories, either incompatible or compatible, right? So let's say, for example, this glycoside and HPMC. So this one is the fingerprint of the glycoside, and this one is the fingerprints of the HPMC, and they are compatible. So we have collected 3,000 such data, right? So it's a rigorous job, tedious job. It took almost 1 or 2 years to collect one by one all the documents we have read it, and we have collected this information. You can see the quality of our data set. This is network graph actually. So basically this shows we have so many drugs with the so many excipients, and we have so many excipients with the so many drugs, in short. So it's not like that we have just one drug and one excipient in the one data set. With a one drug, we have a more than 10. So the neighbor in this network graphing, I have not written, but it is a 15, right? So for one drug, there are almost 15 excipients in our data set, and for one excipient, there are almost 15 drugs. So this is how rich combinations of the data set we have collected and developed. Then we perform the artificial neural network here. So I think today we are not discussing about the artificial neural network in that much detail, but we did it on a Google Colab using the Python language. There is an option and functionality of performing ANN in Orange too. Orange may be the functionality here to perform the artificial neural network, but thoda limited hai. You cannot, let's say, extend it as per your requirement. You need to play in the set criteria. But this is the artificial neural. So if you can see, there are 1,762 inputs. So if you see, if you compare with the design of experiment, so in design of experiment, we do generate the, let's say, kind of polynomial equation, x1, x2, x3, and its relationship with the y. So that's the regression actually. Here, instead of the regression, neural networks runs, but you can compare it. We have x 1762, right? So we have 1762 parameters, different parameters which are used, and the output is a single parameter, whether our combination is compatible or incompatible. Right, so that we performed, and this is its detailing. So once we performed training, right, so we are getting a very good accuracy, 99% accuracy in a training and validation data set. So these are actually the parameters of the machine learning by which you can judge whether your algorithm is enough trained or not, right? Once it is enough trained, then you can use it for the further prediction. So you can see here the training accuracy. We are getting very nice 99%, 91% in the validations. So it's a precision is a very nice 99% in training and validation. Both the data set we are getting a good precision too. So once our model is ready to predict, then we started our actual work. We started to find out different combinations which are not reported elsewhere, and we found so many combination which our model predict as incompatible, and then we are trying to validate by performing DSC, XRD, HPLC, HPTLC, accelerated stability study like all. So this paper actually I just shown you presented such the very interesting example. So see, this is a PCM and vanillin. Paracetamol and vanillin are incompatible as per our model, and we have proven it. Let's say if you see this red line is the DSC. This is the DSC of the paracetamol. So whosoever in takes and can understand the DSC can easily understand. This is the peak of the paracetamol. These two red peaks are the paracetamol peak. This green is a vanillin peak, right? This is the vanillin peak, right? So it's completely. But when we do mix this paracetamol and vanillin, you can see this peak is, let's say, very, let's say, it's totally shifts, right? This peaks totally shifts. So it shows some sort of, let's say, reaction or incompatibility. Then we confirmed it with the FTIR tool. We have stored a sample for a 3 month at a room temperature, and you can see in a let's say, sorry, 1 month old sample, you can see that incompatibility in a FTIR, right? Then we perform the HPLC analysis too, and we lose this some sort of the, let's say, paracetamol here. So this is how our model predicted, and we confirmed. This is HPLC confirmation. Then editor suggested us to add more. So we found another paracetamol with the methylparaben, which is also incompatible. Brinzolamide and this PEG. So it's very interesting. You can see here, brinzolamide and PEG shows incompatible as per our model, which is not reported anywhere. But if you see, this is a fresh mixture of brinzolamide and PEG, and this is just let's say 1 day old, 24 hours after, you may see some sort of coloration, discoloration, yellow coloration, and it is also reflected in the FTIR2, the formation of one sharp peak. So something is happening between two. So this is how we employed AI machine learning to predict the incompatibility. So let's say, how can you use it? So if you go to the GitHub, and just search for my name, Swamiprakash Patel, and you may find this DE interact here, right? So if you click this DE interact, right, it will open so many things. We have actually explained everything here, explained everything, how to use and all. This is 99%, 98% accurate. So I'm also suggesting, if your model is predicting something wrong, you please give us the feedback so we can add that data, retrain it, and make this model more accurate. But here you can see There is one FDP public sharing of DEIP. So if you just click it, sorry, de-interact, sorry. If you click the de-interact, there is a one button. It's called open in Colab, right? So this is what you need to just follow the step, because we yet not created the website. We are soon creating the website. Once we make it a website, it will be easier to access, right? So once you open this, what you can do is you can predict here. How? So you first have to connect. So basically it will connect with the Google server. So basically it will be computed. Our algorithm is already uploaded on it. So it will be computed and it will give the results. What you have to put here, you have to put here the CID. Now, what is the CID? Basically, CID is the PubChem compound ID. So as I said, we have used a PubChem fingerprint here. So you can see if you search, let's say, aspirin, right? So here you can see the CID. The ID of PubChem is 2244. So basically what happens when you add this number, our algorithm goes to the PubChem and then it will find out the chemistry and smile of the compound. Then from the smile, it will convert to the that 808 1-bit fingerprint. So, and then it will utilize it for the prediction. This is how you can add. So if you don't have any compounds PubChem ID, so you can directly add a smile to here. Right? So if you don't have a PubChem ID, let's say if you have a totally new compound, you have developed a new compound and you excipient, so definitely you can find out the structure, you can draw the structure, and there are many tools in internet. You just put the structure and it will generate the smiles, right? So you can use the smile directly instead of the CID, right? So once you do it, you just click this play button and everything will run for a while. So this will start rotating. You have to wait for a while. It will connect first. It is now connected and now it is using the PubChem to develop the data, and then it is using our algorithm. It will download our algorithm one time and then it will calculate. Just wait. Yeah, so it is downloading our algorithm, trained algorithm, which already we have uploaded. There is some sort of error here. So maybe it is happening because Google is continuously updating it every time. So this happening, but once you I will update it. Once you run it, then you have to just click the show result and it will give you whether it is a compatible or incompatible, right? So I think there is an error. Yeah, definitely there is an error because Google is a library, right? So we need to update the code. So sorry for that, but I will correct it, and let's say within a 1 day I will correct it, and once you use it and run it, it will reflect whether it's compatible or incompatible. It will also show you the probability, like just 1 minute. Okay. Yeah, it can show the result here, right? So it will run and will show here what is the status of these two compounds, right? So this is a one work. Then let me show you the another one. So this is how we keep finding, keep learning, and keep doing. I'm not like I'm expert in the AI, but I always fascinated with this, let's say, computer and algorithm. So I learned these languages like Python and all, and started to learn by my own and then develop these works. So the Kuchkost grant that the sir was talking about is based on this work. This is still going on, but we got some sort of the results. So what is it basically? Let me brief you quickly. So let's say if it is a potent drug, very potent drug, 1 milligram dose, even which is a microgram dose. So we know that mixing is a very critical steps while preparing a tablet. So right now what's going on, there are tools too, better tools too, but what we are proposing is totally different. So what we have to exactly do is we have to mix. So एक tumbler में powder घूमता रहेगा, and once it is mixed, protocol is finished, you have to collect the samples. Then you need to perform the quantitative analysis whether you achieved the assay or not. You have to collect the samples, multiple samples from the tumblers, and you need to perform the quantitative analysis. If it is not mixed, then you need to again perform the mixing, and this iterative reactions happens till you get the complete mixing, right? What we are proposing is not actually replacing the quantitative test established by the pharmacopoeia and all, but we are proposing to reduce the unnecessary test, right? So instead of performing the quantitative analysis by HPLC, SPTLC, right? What we are proposing here is we are proposing to take a microscopic photograph, right? So this is the powder mixture where drug is not properly mixed with the excipients, right? And here is a photograph where the drug is completely mixed and this is ready to compress. But you cannot judge just human intelligence, but just by looking at the two pictures, you cannot judge, right? So we are using the technology of deep learning, convolutional neural network technology, right? So basically, just I'm skipping things here. So we have collected the actual data set, right? The photograph of the mixed powder. We have performed the mixing, then we have performed analysis manually, and then we confirm that these powders are mixed and these powders are not mixed. And we have prepared such data set. Then we use this data set for the training of the algorithm, right? So this is our detailed convolutional algorithm. This is again the very core AI component we are not right now discussing, but we have trained our algorithm. And now once the algorithm is ready, you can use a fresh powder mixture to predict whether the powders are mixed or not. So let's say if it says the mixed, then only you should go with the quantitative analysis for the confirmation. When it says it is not mixed, right? So definitely it will save your time. If it is saying that it is not mixed, then you should directly skip the quantitative analysis and remix it. So it will unnecessarily save the time, right? So you don't need to wait until your results arrive at this powder mixture are not mixed. Now you need to mix it further. So based on the just a microscopic picture, so you stop the tumbler, just collect a small powder sample, just put it in a microscope and connect that microscope with the laptop, and this algorithm will tell whether your powders are mixed or not mixed. So this is the one work we did, and we are proposing it to extend it. So we did it for the glipizide. So this is our one of the POC where it completely perfectly predict the uh, powder plant mixing of the glipizide tablet. And we propose that we want, because we have used a very small data set here, so we want to extend it with the large data set. We want to establish this proof of concept with the other drugs, other type, because this glipizide is a crystallines, right? So it's very easy. This is glipizide. Glipizide is a crystalline, so it's very easy for the model to identify it in a sample. So we are proposing that we want to extend this idea with the amorphous drug, amorphous excipients and all. So we are working on it. So that's the one work. And I think few more are there, but I think there are only 15 minutes left. One of the work we did for the HPLC system prediction. So you need to give just information of two drugs that you want to separate, and the system will suggest you system means the composition of the mobile phase and all. So you will have you will have a reference point. So you can start with that reference point and can then optimize. So
Sir,
these are
the
the
one
different works.
question So one question I have seen in the chat box.
Yes,
So
yes.
in that powder, what you have trained with your AI, so
Yeah.
will it be applicable on all kind of powders?
No, 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 sir. No, it's not applicable. It will applicable for that particular formulation. So let's say, for example, XYZ pharmaceutical company is working with the four formulations, right? So what they have to do is they need to develop the database for that for individual formulation only, right? And that's not a big thing. Just you need to take 100 to 200 photographs. That's enough. I think whatever the hours outcome till the date. 100 to 200 photographs are enough. But yeah, definitely they have to do it for the individual formulation. So जो पैक टेक्नोलॉजी है अभी जो हमारा this is also based on this technology, but it works on infrared. So you also have to develop a database for the PAT uh, for individual formulation. So yeah, it is not generalized. It will not work for all the formulation. We are trying that. We also proposed it, but at this point we believe that it will be very difficult to generalize it.
Yes, yes,
But
yes.
for the individual formulation, it's very easy.
Okay, okay.
So this is how to keep learning and keep finding and keep developing the models and work. So I think this is from my end. Uh,
So
if you have,
actually, I'm
yeah.
also getting some many questions like, is there any AI tool for text mining? Means, have you used any tools, AI tools for
Yeah,
your?
for the text mining, I have shown you this orange is the best one. Actually,
Okay,
right now, I actually in the COVID, I learned the Python. So I
okay,
do the coding actually by myself, and it gives you so much flexibility that whatever you want to do. In a software, what happens, you have to rely on the functionality they have provided to you. If you want to
yes.
do certain other calculation can, but if you once learn the Python and then something. So it will be easier. So I actually shifted from the software to the Python, but for the beginners, let's say, Orange is the best tool. It
Okay,
allows to do some sort of basic neural network calculation too.
okay, okay. So thank you, sir, again. And lastly, we are open for question answer round. So if any faculties participants want to ask some questions, you can please raise your hand so that I can give you a chance to unmute yourself and you can ask your question. So any participant who would like to ask any question, please raise your hand. Again, Dr. Ashwini Mishra. So please, you can unmute.
Hello, I'm audible, sir.
Yes, sir. Please ask.
Sir, thank you for such a good presentation. Sir, I need to know how much time it took for you to learn Python.
Sir, very easy. I learned in 1 month.
So,
And sir, this is something like it is not like that you start and it will end one day. Like it's a continuous process. So I started to learn and I understood at the some point that this is the endless process. So at the one point I stopped learning the Python and start executing the my idea. So I started everything on the internet. Like how can I convert the structure into the numbers? How can I convert picture into the numbers? I have seen the papers, then I try to mimic their projects in a Python. Then eventually I understood and start learning it. But yeah, it will take hardly 1 month, or less than
sir,
that.
actually, for the biological student who are into biology and the pharmacy background, there is no such course which is there even in different online platform to learn Python. Most of the time,
Yes,
it is there for computer engineering and
yeah,
others. So, what made you to go in such a depth to learn it? Because once I also tried to do that thing, but since it was only available for
yeah,
engineering,
agreed. I completely agree with you. But
sort this problem out.
again, as I said, as you said, I don't found any course. Basically, it is focused for this engineering people. So as I said, you start from the YouTube पंच अभी आज के टाइम में क्या है कि इवन यूट्यूब पे इतने सारे वीडियोस है अपने वर्नाक्युलर लैंग्वेज में भी होते हैं लोग एक एक क्लिक करके दिखाते हैं कैसे होता है तो आई स्टार्टेड फ्रॉम द स्क्रैच So that's better. Otherwise कोई कोर्स है वैसे, ये कॉर्सेरा वगैरह में बेसिक कोर्स है, यू कैन स्टार्ट विद बिगिनर्स कोर्स आप ढूंढ सकते हैं, पाइथन फॉर बिगिनर्स वैसा।
Sir, at last I need to know how is it really difficult to learn all the script which is there into the Python? How to memorize them?
No, 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 sir. It's a very, say, nowadays ChatGPT is there. You can use the ChatGPT to develop your codes, right? So initially, 2 years back when there wasn't ChatGPT, I do used to type all the codes by my own. When I run, it will go throw me the error. Then I go into and on the, let's say, internet, try to identify what is the error. Then nowadays ChatGPT is there. When we learn it, then it might be the error of single comma that you might don't know. So if you give the chat GPT that I want to do these things, this is the things, I want to write the code about this thing, chat GPT gives you almost 95-98% accurate codes. So that you can use. The most important thing is you must understand what is the code exactly doing, right? Rather writing the code. Let's say if I say my code line is transforming structure into the pumpkin fingerprint, I must understand what exactly that line's doing rather typing the lines exactly. So I think if you merge ChatGPT with the Python, right? थोड़ा बहुत सीखना start करेंगे, तो फिर you don't need to memorize all the codes, right? उसको let's say for example मेरा data है, I need to transform my data from column to row. There is a separate code for it. You don't not need to memorize. Let's say मुझे कुछ columns हटाना है, तो उसके लिए कोड याद रखना जरूर नहीं. You can use the chat GPT कि भाई मेरा ये data है and I need to remove this column. So can you suggest me a code? So it will give the code. The most important thing is you need to check कि वो code exactly आपका requirement वो कर रहा है कि नहीं कर रहा है. That's what I learned.
And one last thing, and it ends over. One AI tool which you suggest as a take-home tool for me today.
You start, sir, with the orange. If you allow me, I can show you very quickly. सर 5 मिनट स्पेंड कर सकते हैं सर?
Yes, sir. Yes, sir. We have time.
So yeah, a few years back, I was also finding the softwares. So you can also start with the orange as I shown you today. Orange, there is the functionality of the neural network. Yeah, these are the models, and yeah, this one is the neural network, right? So you just go to the website of the orange. You first start to understand what is artificial neural network, what is the basic fundamental of artificial neural network, because the unit calculation, the most minute calculations behind every AI models is a neural networks, yeah, complex neural networks, right? So you need to first understand what is neural network, then go to the orange websites. They have given a free data sets to try understand how this neural network works, try to predict, try to train it. So this is how you can start with. I suggest you start with the first understanding ANN and go with the orange tool where they have given a neural network. Basic functionality, there is not much because see here they have given only 4 functions. In neural network there are so many functions, but at least you can start with these 4 functions and these 3 solvers. They also give the basic data sets to practice. तो एक छोटा सा डेटा सेट लो, उसके ऊपर न्यूरल नेटवर्क लगाओ, फिर उसको समझो व्हाट्स गोइंग ऑन हैपनिंग. वंस यू अंडरस्टैंड वेल कि भाई ये तो ऐसा हो रहा है, राइट, देन यू स्टार्ट understanding its coding and then extend it.
Thank you, sir, for your explanation. So now I like to request Dr. Geeta, you can please unmute and you can ask your question.
Thank you once again for your nice presentation, sir. Sir, how it's applicable in chemical synthesis, sir?
That's total depends on your creativity and idea. There are so many numerous applications. ऐसा मैंने कोई फील्ड देखा नहीं जहाँ पे अभी एआई का एप्लीकेशन नहीं है. मैकेनिकल में भी बहुत सारे एप्लीकेशन. आई कैन नॉट डायरेक्टली राइट अभी भी, आई कैन नॉट जनरेट द आइडिया राइट नाउ, बट आप थोड़ा लिटरेचर सर्च करेंगे तो मिल जाएगा, लाइक फॉर एग्जांपल कोई स्ट्रक्चरल डिफॉर्मेशंस हो रहा है, या फिर लेट्स से बहुत सारे ऐसे मशीनरीज है जिसके पार्ट्स जो है वो अगर उसमें से बिगड़ रहे हैं तो द सिस्टम कैन अर्ली डिटेक्ट इट। उसके साउंड के बेस पे इसका उसका जो परफॉर्मेंस आ रहा है, तो सेंसर विल कलेक्ट द डेटा, दैट कलेक्ट विल डेटा विल गोज टू द मॉडल, एंड मॉडल कैन वेल बिफोर द टाइम कैन प्रेडिक्ट कि ये पार्ट वर्न आउट हो गया है, यू नीड टू change it before complete breakdown. So there are so many applications, so many. But you need to first, as I said, learn this ANN and all, learn programming, and simultaneously keep reading. There are so many small projects people do discuss and let's say elaborate on a YouTube and all. So you should study that, how they have performed. You try to execute a small project. So eventually laterwards you will have a better idea कि मैं ये भी कर सकता हूँ, फिर इससे तो ये भी कर सकता हूँ.
Sir,
Kind of.
whatever
This
the
is how I learn.
information, yes, sir. Whatever the information we get in chat GPT, we can follow, sir, 100%. I mean, we
No,
will
no.
make the changes.
Not 100%. That's why that's a different topic, but generally we use ChatGPT for as a search engine. So don't use ChatGPT for a search engine. As I said that I use ChatGPT for developing my code. So I give instruction to the ChatGPT that I want to do this thing, Please write a code for me. So it write a code. I do not go to the ChatGPT and directly search like what is artificial intelligence, what is what is this. For that, better search engines are there.
Thank you.
So चैट
Yes, yes.
जीपीटी का आंसर 100% तो नहीं होता है, बहुत सारे मिस्टेक्स होते हैं उसमें.
Thank you, sir. Dr. Shravan, you can unmute and you can ask your question.
Good morning, sir.
Good
Is
morning.
it audible, sir?
Yes,
Hello?
sir. Yes, sir. Yes, sir.
Sir, can you suggest any AI tool for pharmaceutical people? So to know the drug and excipient interaction that you said now.
There are, but वो सारे पेड होते हैं, लाइक पर्किन एल्मर के या फिर ये सॉफ्टवेयर्स आते हैं उसमें टूल्स होते हैं, बट दे आर वेरी पेड कॉस्टली सॉफ्टवेयर्स. For are you asking for this drug excipient prediction?
Yes, sir. Yes.
हाँ, तो कैम ड्रॉ वगैरह में है कुछ टूल्स है, बट दे आर पेड वन. I don't know how they actually develop, but we have developed our own and we capped it on open source, so anybody can open it. I have shown you there is minor error. This happens every time when Google updates their servers, but I will update it and it will be ready. So you can just go to the GitHub, search my name, and you can find out the interact, and it will be easy for you to predict incompatibility for your formulation.
Thank you, sir. Now, Dr. Jayalakshmi Venugopal. Dr. Jayalakshmi, ma'am, you can please ask your question.
Sir, I want to know whether this Python algorithm can be used for genotypic analysis for drug response.
Yes, yes, yes. There are so many papers. You can.
If suppose we are not able to do it ourselves by learning Python, or any other consultancies are there which can help us
Yeah,
to
the best way right now I'm doing is I just let's say I just stuck with the codes right now, means I don't want to write the codes my own. So because I understand what's going on in my code, right? So now what I do, I made a collaboration with the students here in my campus in a field of AI. So I have exactly idea what to do. They have a very good skill to draft the codes. I have a skill to read that codes, what exactly going on. So what I do, I make a collaboration. I make them understand that this is what I have to do. You please draft the codes and let's start working. Then I will just check the codes. Is there any error or not? Or, this is how I am working. So
Okay. Can you
making a
suggest
collaboration with the computer guys is best way.
Can you suggest any reference for us so that we can also take help for learning
Yeah,
between the?
definitely. I am open. If you have any idea in my campus, I have identified a very good faculty and students who are working with me, so they can also collaborate and we can discuss afterwards.
Sure, sir. Thank you. So I'd like to know from PP Savani University to give a sir's reference so that we can be in contact with them.
Sure,
Yeah, sure,
sure.
sure.
Yeah.
Thank you, sir.
So, I think we are already done. Anyone is like to ask any question? Okay, Dr. S. Prasanthi, you can please unmute. Dr. S. Prasanthi. Just 1 second. Yes, Dr. S. Prasanthi, you can ask question.
Yes, sir. Good afternoon, sir. This is very
Good afternoon.
informative session. Exactly, I want to know how these artificial neural networks differentiates from factorial designs, sir. How we can
Differentiate.
differentiating from factorial designs. How factorial designs are for optimization. And these neural networks, how this will help us in development of the drug delivery systems.
Uh, Basically, see, what is artificial neural network do? Just let me open up the figure so you can understand. See, so basically, yeah, I have not shared. Just 1 minute. So this is actually a mathematical calculation. So let's say this one, right? So basically what happens here? So these are the inputs, right? So what I exactly do, I prepare an exact data set. As I said, let's say how neural network works in a drug delivery. So let's say I want to develop a system which can predict for example, topical penetration, how much drug will penetrate. So I will prepare a data set, known data set. Like these are the different combination of the formulation. And for that, we are getting this much of the penetration, for example. So this is the known data set. So now what we do, we feed this known data set where input is known, output is also known. We call it as a label data set actually. Right, so we give this data set to artificial neural network. Now, what is basically artificial network do? It will calculate the certain values. We call it as a weights. That's what exactly I just referred a few minutes back, that you need to first understand the artificial neural network. So it actually calculates the values, this weight, and it actually not calculate, it adjust, right? So you need so much computational power. It continuously in a fractions of seconds is change so much values, and it keep changing the values till you get the exact output for your exact input, right? So let's say, for example, you have given 1,000 inputs, and for that you have 1,000 outputs. So out of 1,999, let's say 1999 percentage exit output आ ना जाए, तब तक ये values को change करता रहेगा. Once it achieved it, right? So it may take, let's say, depending on your data, it may take 1 or 2 or maybe day or maybe more than that. Let's say ChatGPT आपने देखा होगा, तो it's a so huge data set कि उसमें महीने लगे थे to calculate that exact value. So once you calculate it, right, and once your model is ready, right, so for 1,000 inputs, it predicts its output 99% accurately. Right now, it is ready to take the unknown inputs. Now it will take unknown inputs, and for it will predict the output which must be accurate, because उसने 1,000 data के लिए accurately train किया हुआ है. Larger the data set, larger, more accurate your model. So this is how ANN model works. But the question you are asking, how it helps in particular, let's say, idea. For that, you need to understand what data I should create, right? For the floating tablet के लिए, let's say for example, जो कितने time में float हो रहा है, right? तो you need to devise कि क्या क्या major input data होंगे. Let's say उसका excipient, उसका composition, उसका amounts रहेगा, media rahega, let's say for example, and let's say floating time you want to predict. So you need to prepare thousands of tablets, right? Yeah, you need to collect the data from the available good reputed journals, and then you need to feed this data to neural network. Right? This is how this things works.
Thank you, sir. I hope I will get the answer. So this is really a wonderful session. And I thank you again, Swamiprakash sir, for such a profound and engaging session. So your insights into AI tools and their transformative potential in patenting are invaluable. So, sir, please accept this certificate. Just give me a second. So, this accept this certificate of appreciation from PP7 University as token of our gratitude and
Thank you
recognition of
very much, sir.
your valuable contribution. So thank you
Thank you
again,
very
sir,
much.
for your time and for accepting our invitation and to share your insights for this EFTP. Thank you
Thank you
again,
very
sir.
much.
Thank you, sir.
Okay.
Now all the participants are requested, please kindly fill the feedback form, which is already floated in the chat box. I request all the participants kindly fill the your attendance and feedback, which is floated in the chat box. Swamiprakash sir, if you want to share your mobile number in the group,
Yeah, I'm just putting my number on a, let's say, in a chat, right? Is it okay?
yes, sir, yes, sir. You
I'm
can
just
share
on the chat.
the okay, you can share your GitHub ID also if you want in the chat box.
Yeah, just 1 minute. I will
Yeah.
put it in. Just paste it. Yeah. Yeah, so I just pasted it in a chat box.
Okay, okay, sir. Okay, sir. Thank you, sir. Everyone, you can please switch on your camera. So at last we can have some photo at the end. Everyone, please switch on your camera. Thank you, everyone. Thank you for your time. So tomorrow is the last day. I hope you have enjoyed the session today. And by tomorrow, we'll again join. We'll learn something new, AI and ML in pharmaceutical science. Thank you, sir. Thank you, everyone.